Well, good day to everyone joining us and welcome to today's X-Talks webinar. Today's talk is entitled Advancing Alzheimer's Clinical Trial Endpoints, Imaging Biomarkers, COA Challenges, Movement Measures, and CV Risk. My name is Ryan Muse and I'll be your X-Talks host for today. Today's webinar will run for approximately 60 minutes, and this presentation includes a Q&A session with our speakers. Now, the webinar is designed to be interactive, and webinars work best when you're involved. So please feel free to submit your questions and comments for our speakers throughout the presentation using the questions chat box, and we'll try to attend to your questions during the Q&A session. This chat box is located in the control panel, which is on the right-hand side of your screen. And if you require any assistance along the way, you can contact me at any time by sending a message using the same chat panel. At this time, know that all participants are in listen-only mode, and please note that the event will be recorded and made available for streaming on xtalks.com. At this point, I'd like to thank Clario, who developed the content for this presentation. Clario is a leading healthcare research and technology company that generates the highest quality clinical evidence for pharmaceutical, biotech, and medical device partners. Clario's science, technology, and operational experts have helped deliver over 26,000 trials and contributed to over 800 regulatory approvals in more than 100 countries. Now I'd like to introduce our speakers for today's event. Our moderator today is Dr. Todd Rudo, the Chief Medical Officer at Clario, with over 20 years of clinical cardiology and pharmaceutical research experience. He provides medical and scientific leadership across the organization. Also joining us today is Chris Conklin, PhD. He is Director and Neuroscience within Clario's Medical Imaging Group, who specializes in applying a range of MRI modalities within clinical trials. Dr. Pratik Verma is Director, eCOA Science and Consulting at Clario, overseeing eCOA design, investigator training, and outcome analysis, with particular focus on neuroscience indications. Dr. Vic Patel is Vice President and Chief Medical Officer of Cardiology at Clario and oversees cardiology consulting and ECG Holter core lab services, in addition to the cardiac biostatistics and medical writing groups. And Adam Jagodinsky, PhD, is Principal Scientific Advisor for Precision Motion at Clario, where he focuses on supporting sponsors to implement wearable sensor solutions in their clinical trials. But without further ado, I'd like to hand the mic over to our moderator, Dr. Todd Rudo. Todd, you may begin when you're ready.
Okay, great. Well, thanks for the kind introduction, Ryan. Good morning and afternoon, everyone. Thank you for joining us. As Ryan said, my name is Todd Rudo. Glad to have the opportunity to host this interesting and really quite packed session today about digital endpoint strategies in Alzheimer's disease studies. Over the next 45 minutes or so, we're going to take you through a range of clinical trial endpoint solutions, highlighting the role that innovative technologies can play when a successful development within a successful development program. You can go to the next slide, please. So prior to today's webinar, we launched a poll on LinkedIn, which many of you responded to. You can see about 140 or so. We asked, what is your greatest concern for your Alzheimer's development program? The highest percentage of you were concerned about difficulty demonstrating efficacy, which very commonly comes up because these are such long studies and it's a slowly progressive disease. Also of major importance to you was a particular challenge to this population compared to other clinical trials, and that's whether the cognitive impairments of the participants will have an impact on trial success. Underlying this typically are concerns with recruiting and retaining patients in this state, or concerns for collecting certain endpoints accurately, such as patient-reported outcomes. A smaller percentage of you were concerned about the complexities of imaging modalities in this space, which of course we'll hear more about today from Chris, and along there were also concerns with a population at high risk for cardiovascular disease could have impact on study conduct and data interpretation as well. On the agenda, on the next slide, after I give a brief introduction to level set us all on the fundamentals of what is Alzheimer's disease and where we are today on the journey to developing better treatments, our panel will then each take you through some elements here. So imaging solutions for eligibility, safety, and drug efficacy, digital gait and movement assessment, common endpoints in eCOA, and specifics of how to best deploy them within an Alzheimer's program, and where cardiovascular testing should be deployed, given the high cardiovascular risk these patients also come into the clinical trials with. So what is Alzheimer's disease? I can go to the next slide. It's a type of dementia, and it's the most common form of dementia. And it results from a loss of neurons or the brain cells essentially over time. It's therefore considered a degenerative neurologic disease, shares that category with diseases like Parkinson's or ALS or multiple sclerosis. What's especially interesting, though, is that changes in the brain begin to develop 15 or more years ahead of any symptoms of cognitive decline. The prevalence is quite high, over 60 million people globally that are affected. And while substantial effort has gone into understanding risk factors, this is still fairly nebulous. Age is at the top of the list in terms of risk factors, but also family history, female sex, cardiac and cerebrovascular disease are also contributing. There's been some evidence for genetic susceptibility. No singular gene has been identified as a sole cause, though. It's largely a clinical diagnosis, and it's exclusionary, meaning that other types of dementia need to be ruled out first. Certain imaging findings can help be supportive of the diagnosis, though, particularly including volume loss, the presence of amyloid plaque buildup in the brain, and something called tau deposits that we'll hear more about today, and that's a protein accumulation in the brain. The symptoms initially are very non-specific, but they do typically include early trouble with things like word finding, memory, problem solving, mood and behavior changes are common even in early disease as well. And then as it progresses, very profound memory impairment becomes the predominant feature, along with progression in the severity of the other symptoms I mentioned, at which point the patients begin to struggle with and really need support for the most basic activities of daily living. So things like bathing, getting out of a chair, getting out of bed, dressing, using the toilet, even feeding, all of that becomes difficult as the disease progresses. On the next slide, I'm just kind of focusing, okay, so what can we do about it? Well, first, I'll mention the non-pharmacologic management, which really focuses on what may be contributing that we can modify, even if the underlying disease process can't easily be stopped. So this includes addressing any undiagnosed sleep disorder, addressing cardiovascular risks, encouraging an exercise routine, and so forth, as indicated here on the left. For medications, though, back in the late 90s, there was so really 25 years ago now, a drug called donepezil or Aricept came onto the market. This is in a class of drugs called acetylcholinesterase inhibitors, which boost the levels of a neurotransmitter in the brain called acetylcholine. And they slow the progression of some symptoms of Alzheimer's disease. They do not actually improve the underlying disease itself, and they do have some cardiac and gastrointestinal side effects to be aware of as well. But certainly were an important milestone in efforts to find any treatment at all, for Alzheimer's. There's another class of drugs called NMDA antagonists or inhibitors. These are reserved for more significant or advanced symptoms of Alzheimer's, also associated with some improvement in symptoms, but yet again, not really targeting the underlying cause. More recently, there were 2 approvals last year you might be familiar with, drugs that specifically target the amyloid plaques that I mentioned. This is especially important because now we're talking about treating the underlying mechanism of Alzheimer's, not just the symptoms of the disease. And so hypothetically, at least if you can treat that underlying mechanism in high-risk individuals and treat it very early, you could potentially delay or even halt the onset of clinical disease altogether. So that's why this is particularly exciting. You may have actually even seen this week, there was an FDA advisory committee that voted favorably on a 3rd medication in that same class. Similarly, drugs are also under development targeting soluble tau, which is, as I said, a protein deposit, which is another contributing factor to this neurodegeneration process. Nothing on the market yet in that mechanism, but certainly under development. With that, I'm going to turn over to Dr. Conklin to talk about the different innovative imaging modalities that are relevant in Alzheimer's disease, including those to aid in the diagnosis, to confirm eligibility for a study, imaging as a biomarker for evidence of efficacy, and then also the role for imaging to monitor safety, particularly with amyloid-directed therapies, and a unique risk that we've discovered that's associated with them. So Chris.
Thank you so much, Todd. I appreciate it. So just to start, while many of you may already be familiar with the imaging expertise we have here at Clario, I just wanted to try and provide a very high-level overview of imaging within the Alzheimer's disease landscape, and I'm certainly going to try my best to condense it into a 10-minute window for everybody here. Next slide, please. So as Todd already mentioned, there are two key neuropathic features that are seen in the brain of Alzheimer's patients, which are amyloid beta plaques and intracellular neurofibrillary tangles that contain hyperphosphorylated tau proteins, as he indicated. If you want to take a more clinical perspective, this disease tends to manifest through progressive memory problems, which leads to what is known as mild cognitive impairment, or MCI, which ultimately then cascades into dementia, and unfortunately, ultimately with enough time, death. But what is becoming clear is that these disease-modifying therapies must engage biological targets, ideally long before sort of this pathophysiologic onset that leads to dementia. And so to aid in this pursuit, it's logical to try to define Alzheimer's disease within a feasible biological framework and an ability to try to enhance our understanding, characterization abilities, treatment and prevention methodologies within this horrible disease. Next slide, please. So currently, the accepted biomarker-based research framework for Alzheimer's disease is called ATN, amyloid tau neurodegeneration, as you see here. And so it is broadly categorized into just 3 groups of biomarkers. And as this talk progresses a little bit, I'd like to try to expand that classical thinking to be able to target certain biomarkers that may manifest even earlier within, the disease sort of state, if you will. So as you can see here, for the amyloid case, this is, again, aggregated beta amyloid proteins and any associated pathology therein. There are a myriad of methods that we can use to test not only the extent and severity, but also the presence, through CSF measures like AB42 or even a ratio AB42 to AB40. That ratio itself may allow one to normalize the beta amyloid levels across different patients with, the potential for maybe even being able to predict patient trajectories throughout the course of a clinical trial. We have the proclivity, which is a blood test. This is accessible, it's radiation-free, it's minimally invasive and cost-effective, particularly for eligibility and enrollment purposes. They also have it available for APOE status as well. And then obviously there's the molecular imaging approach, which is amyloid PET, which for my money is sort of the gold standard for being able to locate the extent and severity of amyloid depositions within the brain. Moving quickly to tau, they have, again, similar battery of methods there from CSF, looking at phosphorylated tau, blood tests for p-tau217, and then also tau PET molecular imaging techniques to look for, again, localized tau deposition. And then moving on to neurodegeneration or neural injury, we have the total tau measure, which is a CSF measure, FDG PET, which can help measure cerebral metabolic rate of glucose. So you can think of it sort of as a measure of neuronal activity. So these metabolic changes tend to precede any clinical manifestations of said disease, in this case, Alzheimer's. And then we jump into, my bread and butter, which is the MRI component, in this case, anatomical MRI, looking at things like volumetric change or atrophy over time. Next slide, please. Before digging any deeper into this, though, and some of the biomarkers, I feel it is absolutely critical to talk about the safety component within AD. A lot of these anti-amyloid therapies tend to lead to what are known as amyloid-related imaging abnormalities, as you see here. Won't have too much time to go into this too much detail, but ARIA is believed to be caused by an increased vascular permeability, particularly due to inflammation, which is typically a result of increased amyloid plaque burden within an individual. And we at Clario have extensive experience with this abnormality and played a crucial role in terms of the development of these scoring paradigms on a lot of the therapies that have come out to date, which you can find more on at understandingaria.com. But I think the take-home message here is that both of these ARIA flavors, both the edemic as well as the hemorrhagic, can be visually assessed and characterized with standard imaging sequence within most imaging facilities around the world. So FLAIR, which is a fluid attenuated inversion recovery sequence for the edema characterization, and then the T2 star, which is a bread and butter gradient echo sequence, which is absolutely phenomenal for visualization of blood products. And there are both 3 and 5 point scales, as you see in the upper left-hand corner for ARIA. The ARIA E for ARIA H, you know, if you want to look at a 3-point scale, which I know very well, just to give you an example, you know, it's typically less than 4 microhemorrhages that are seen there. 5 to 9 is sort of number 2, and then anything greater than 10 with the presence of superficial siderosis is you know, probably severe. And the good news is that, you know, on the edemic front, you know, halting the administration of the drug will actually reverse these effects for the most part. But again, please go to understandaria.com if you're interested in finding more about this, because I'm just a physicist, not necessarily a clinician, so I'm sure a lot more detail is contained therein. Next slide, please. So jumping quickly back into molecular imaging, PET, both amyloid and tau, are fantastic tools within the context of an Alzheimer's disease trial because they can be used for both eligibility as well as efficacy. So in terms of amyloid, when looking at the visual assessments, you know, there are some key regions here where we start to notice this, you know, the temporal lobe, frontal lobe, parietal, the cingulate and precuneus. You know, these regions are also typically where you will calculate something called the SUVR, which is the standard uptake value ratio, while using the cerebellum as the reference region. Looking at tau, this uptake tends to occur initially within the medial temporal cortex. And then for those that may not be necessarily too familiar with this, there are what called Brock stages. There's 6 different stages. So just in stages 1 or 2, these NFTs or neurofibrillary tangles tend to be isolated to the entorhinal cortex. As we progress to stages 3 and 4, they begin to infiltrate the limbic system, such as the hippocampus. And then in stages 5 to 6, we begin seeing more neocortical involvement. So what has been shown is that moderate levels of amyloid are required before tau uptake can be detected outside of the entorhinal cortex. So you're starting to see sort of this cascade of different events. And from an eligibility perspective, you know, there are quantitative centiloid cutoffs that can be used. For amyloid, it's 20.1, and currently for tau tracers, there is a harmonization effort underway called the Centaur, which hopefully will be validated shortly. Next slide, please. So real quick here, you can see that based on the positivity or negativity of amyloid, tau, and or neurodegeneration, based on some of the techniques that we've already discussed and associated with sort of the cognitive or clinical scaling, you can start seeing, you know, sort of a level to predict and sort of see what state the patient is in. And really, I think the take-home message here, as you see on the plot on the right, when plotted at, you know, sort of the age and against the preclinical Alzheimer's cognitive composite score, you can see for those individuals that are both amyloid and tau positive, which is the orange curve, that's not a good trajectory to be on. So it's pretty much safe to say that once you have both of those positivity values for amyloid and tau, that the outcome is not good, which is why we need to start targeting this earlier and earlier. Next slide, please. So quick modification here. This is a modification of the classic Jack curve showing sort of how these biomarkers change with time. And just one thing I want to talk about is you can see the amyloid marker in the green curve there. And then there's this red curve that I really want to try to focus on given the limited amount of time that I have here. And those are the functional and metabolic markers. And you can see that they start showing a percentage of impairment and a degree of change even before MCI takes place. And this is really what we're trying to do. It's come up with better eligibility and screening techniques to be able to recruit patients for these clinical trials within that window and then leverage more advanced quantitative biomarkers to help. So if you just advance this so the next little figure pops up for me, please. Just taking a more granular look here into this functional metabolic curve, we start seeing that there is a systematic staging of biomarker evolution, which can be detected, as I said, prior to clinical symptoms. In this case, you know, mild cognitive impairment. We have early MCI, late MCI, as you can see on that x-axis there. And the thing I want to highlight here is if you look at that orange sort of gold curve there, that's vascular changes, which can be measured in MRI through arterial spin labeling. The gray curve there is functional MRI, looking at functional connectivity within the brain. And then there's other metabolic scores and things that can also be taken a look at, which will show a change, which may be representative of an individual being on a specific course in their progression. Next slide, please. So just to wrap up, my take-home message here is that, you know, really it's not one of these markers, CSF, blood, clinical score, PET, MR, whatever, you know, biomarker that's going to sort of paint the entire picture here in order to fully characterize and predict a disease and, you know, therapeutic efficacy, disease state within an individual. It's really sort of the multimodal integration of all these different orthogonal data sets together that's going to truly paint the entire picture and to really give us a true idea as to the state of a given patient and also the therapeutic efficacy based on a compound that's being tested within a clinical trial. And you can see here, there are a couple biomarkers that I'd like to point out. You know, we talked about amyloid and tau on the molecular imaging front. Functional MRI is very widely available despite the misconceptions. I could have a whole separate hour-long talk about each of these individually to tell you about the availability, the way in which they're acquired, but unfortunately, I need to wrap up within the next 30 seconds here. ASL is used for perfusion measures, calculating something called cerebral blood flow. It's a quantitative MRI method. That is a more heterogeneous distribution in terms of availability across the globe. You know, we're looking probably about 50% of sites that have those capabilities. And as we move away from the 2D into the more 3D pseudo-continuous ASL techniques, which are more accurate, more reliable, that number begins to shrink a little bit more as well. DTI is becoming more and more ubiquitous, a great way to look at structural connectivity and white matter integrity, as we know that also degrades within neurodegeneration. And then QSM, something that is emerging and does show, obviously, iron plays a huge role in most neurodegenerative indications, and being able to accurately quantify that without, you know, the actual signal blowout that you see in traditional GRE sequences. This does allow us for precise visualization and quantification of iron within the brain, which may help, again, sort of elucidate any sort of therapeutic treatment and or where they may be along the line of that pathophysiologic cascade of neurodegeneration. So it's really the integration of all this that is going to paint the whole picture. And we at Clario have the experience to be able to do this and have widely deployed all these in the context of multi-site, you know, global clinical trials. So with that, I do apologize for going over a little bit. I'm going to hand it off to my colleague, Adam, who's over in Precision Motion. Thank you all so much.
Thanks, Chris. You can move to the next slide, please. All right, so as Dr. Rudo mentioned in the intro, mobility, gait, and balance deficits are present in patients with diminished cognitive function. This is a result of overlapping cortical networks in the central nervous system. And this is particularly significant because these gait and balance impairments can lead to falls, disability, and in general affect the overall quality of life of patients. And so in addition to that, a growing body of evidence is providing some good insight into the utility of gait and balance measures as clinical tools for screening cognitive decline and dementia. And so moving forward, we'll touch on some of these points in greater detail. So this model here illustrates the interrelationship between cognitive and gait impairments that occur with neurodegeneration. And so on the figure, the white arrow here describes the interrelation between cognitive impairment and gait impairment, as well as the interrelation between dementia syndromes and falls and fractures. So the gray arrow indicates the spectrum of cognitive impairment, which is shown on the top, as well as mobility decline shown on the bottom, that occurs with aging and neurodegeneration and leads to increased risk of dementia and falls and fractures, respectively. And so the dotted arrow, mainly the one starting on the bottom, and shows gait mobility impairments pointing to dementia syndromes, is an indication of the predictive capacity of gait mobility measures along the spectrum of neurodegeneration. Next slide, please. And so here we're showing that gait is a complex behavior, and it consists of several key areas or domains, including pace, temporal or rhythm, variability aspects, asymmetry, stability, turning, and balance as well. And particularly the pace and rhythm domains or temporal domains have been identified as key domains that are sensitive to mild cognitive impairment and Alzheimer's disease. And these domains can be assessed using instrumented walking, balance, and mobility tests, which we show in the next slide here. So here are, this figure shows opal wearable sensors that can be implemented during prescribed gait, balance, and mobility tests under various conditions. For example, a dual task paradigm can be used to target the interplay between cognitive and gait domains. And so the opal technology is suitable for trials and have been demonstrated to have utility to capture both accurate and precise and reliable movement endpoints in neuroscience trials. So in the next slide, we look at some data from a recent study that uses wearable sensors to capture gait and balance outcomes in individuals at risk for cognitive impairment. So this top left model illustrates how individuals with Parkinson's disease, specifically who carry the GBA gene variant, are disposed to cognitive and movement impairments. And the cognition is shown here as a mediator that leads to more enhanced or exacerbated gait and balance impairments. And on the bottom left, you can see the study specifically had participants complete both walk and balance tests while wearing the Opal wearable devices to objectively assess the domains of gait and balance. And the results shown on the right are the correlations between tests of cognitive function for each of the domains of gait and balance, which are shown in the rows. And specifically here, the length of the bar indicates the strength of the correlation. And so the executive function tests shown on the bottom were most correlated with the pace and turning domains or measures of gait. And on the top, we look at the attention and visuospatial function. Those are most correlated with gait variability and balance measures for this particular study. So it highlights kind of the specific links between different cognitive domains and as well as the movement domains, gait and balance domains. Next slide. So this slide is looking at sort of highlighting a literature review from 2016 that looked more broadly at gait domains associated with cognitive function in Alzheimer's disease and mild cognitive impairment populations. And so several of the review articles here point to measures specifically in the PACE domain of gait that show association with changes in global cognition, executive function, attention, as well as progressive decline of cognitive function. And you'll see also the McKay paper showed association between gait rhythm measures and cognitive function as well. So this review points to some of the scientific evidence that links specific domains to discrete cognitive functions. And in the next slide here, we'll look at some more data from a study that assessed the value of instrumented gait analysis in individuals with mild cognitive impairment and Alzheimer's disease. And so this study involved both neuropsychological assessment and motor assessment, which included instrumented gait analysis in control group, mild cognitive impairment group, and Alzheimer's group. And so the two tables on the left show gait data from both a single and dual task paradigms. And so the PACE measures showed sensitivity here to Alzheimer's disease in both of those paradigms, single and dual task conditions. But in the dual task condition, the PACE measures were sensitive to mild cognitive impairment and Alzheimer's disease. And on the right-hand side, this table shows the PACE measures of gait speed and stride length were both correlated with measures of global cognition and attention. So this provides some insight into the utility of using instrumented assessments, gait assessments for distinguishing gait patterns in mild cognitive impairment and Alzheimer's disease, and particularly points to dual task conditions, which may be useful when assessing gait impairments associated with cognitive decline. Next slide, please. So just a quick summary. Gait is complex and it involves a distinct relationship between gait domains, specific gait domains, and discrete cognitive functions. And gait domains, or excuse me, gait measures, primarily in the PACE domain, correlate with measures of cognitive function. And these are sensitive to both mild cognitive impairment and Alzheimer's disease. And then finally, objective gait measures show potential as clinical trial endpoints to supplement traditional outcome assessments for mild cognitive impairment and Alzheimer's disease. And to segue, I will pass to Dr. Verma. Thank you.
Thank you, Adam. So as you listened today with some of my colleagues, you know, when you're designing an AD program, what are the challenges that you face? And if you go to the next slide, we know AD progression not only leads to deterioration in patients' cognition, but also functional autonomy and motricity. From endpoint perspective, we see a gradual shift in use of more and more measures which explain the holistic change in patient leading to deterioration in cognition, functional abilities, quality of life, and objective biomarker like motricity. If we go to the next slide, we'll see what are the challenges when you're designing studies in neuroscience. From endpoint perspective, looking into if the endpoint is able to capture the subtle changes of or early disease stage, floor to ceiling effect, is the instrument has limits to its ability to detect temporal improvement or decline, practice effect, poor design leads to learn effect, may lead to poor data integrity. And some of this has been published by some of my ex-colleagues, particularly looking into poor solution design of endpoints like ADAS-COG and MMSC. Paper versus eCOA. We definitely know that, eCOA gives us the meaningful data. Is paper still relevant? Can we get clean data using paper scales? And is the eCOA captures the changes in patients similar to a paper? So these are some of the findings where we have studies from Campbell and others where they point out implementation of eCOAs and equivalence to paper. Rate of variability. We know that poor consistency in rating leads to drug not meeting the study objective, and raters do not have good inter-rater reliability. We are bound to see this issue crop up. Recruitment challenges cause havoc, and if the recruitment challenges are not met, then there won't be timely completion of the study. And if this is compounded by poor patient selection, then study won't meet its objectives. So what does the regulator say? So let's look in the next slide, please. So both US and European regulators cite use of multiple endpoints to holistically analyze the disease progression. However, FDA goes further, stating that we should consider having endpoints and biomarkers, which is a pretty broad term. And, some of my colleagues like Dr. Conklin and Adam spoke about, imaging biomarkers, gait and balance biomarkers, but also there is blood and plasma biomarkers. So, it is a broad term, and then we have to analyze, what should be about broad, biomarker, which should be used in along with the endpoint, which identifies early stages of disease where functional impairment remains intact. So moving to the next slide. So we scanned through clinicaltrials.gov using a bot to determine changing endpoint landscape in the last couple of decades. And we saw an increase in cognitive, functional, and global instruments like ADAS-COG, CDR, CIBIC, by more than 20% over a period of, from 1993 to 2023. However, we see a gradual change towards MCI and pre-AD studies in a bid to identify a patient at risk for developing AD. And if we move to the next slide, so what we have learned and how can we improve it? So to address some of the endpoint challenges I talked about earlier, let's go to the next slide. We should ensure that, we should have good inter-rater reliability by rater training and testing, have automatic scoring with eligibility reports on devices, and that should help us address some of the challenges in identifying, issues with the endpoint, built-in logic to address data anomalies in real time, decrease patient burden by designing ePros with patient in mind, address data issues with central monitoring, utilizing audio or video capture, and get early insight into the study using statistical and data analysis. If we go to the next slide. So here are some of the examples which I'll be citing in the next 3 slides, explaining how we can address some of the challenges I highlighted earlier. So to address inter and intra reliability, we should employ training to the study startup, followed by close monitoring for inter and intra-assessment consistency, and this can be done using central monitoring, as I talked about, utilizing audio-video capture and getting it centrally reviewed, and also data analytics throughout the life cycle of the program to determine if there are any data anomalies and address it on a timely basis, so that we do not encounter issues from a high enrolling side, which would affect the overall data in the study. The next slide gives us another example of intra-assessment consistency. Check in real time on eCOA devices, for example, ADAS-Cog, spoken language versus comprehension of spoken language. And these can be built in real time, and the investigators, while they are scoring the instrument, they would be flagged up of disparity between the two responses. The next slide is another example of a clinician of CDR, where intra-assessment real-time alerts can be built in to address community affairs scoring, alerts to address any scoring disparities which is caught early on and which might perhaps affect the eligibility of the patient in the program. The next slide gives us an overview of intelligently designed diaries. So, sleep diaries which eliminate double data entry and collects clinically meaningful data associated with sleep duration, quality, and breaks in sleep, helps document patient sleep pattern and eliminates data anomalies. So if we go to the next slide, which would be a summary of how we can address the challenges in clinical endpoints in Alzheimer's disease. We should be looking into appropriate timing and format, life of the trial, considering when and how the endpoints will be used throughout the trial. Should the endpoints be changed based on maturity or disease progression? Shifting between an observer-reported outcome measures to a patient-reported outcome measures, or vice versa. For example, long-term, we might see that patient might be able to report their symptoms, but as they progress in the study and the disease progresses, they might not be able to be able to report on what they are going through. Patient completers and caregivers provide support structure to patients and to their caregivers to be able to document their assessment properly. Loss of ability, as I talked about, clarify how the assessment will be handled when the patient can no longer complete them independently. Data integrity, looking into that the caregivers do not complete the assessment on behalf of the patient without proper guidance. Patient engagement, align with the goals, understand how endpoints fit within the broader study objectives, capture relevant data while enhancing the patient experience. Best practices, and I talked about earlier as well, looking into training of the patient and the caregiver, as well as your study staff on how the eCOA would be assessed, and the usage of the eCOAs. Clear instructions provide, making sure that not only your study investigators but also patients and caregiver have clear instructions how to complete the instrument. Patient-centric design with patient in mind, and adaptive approaches, making sure that the endpoint is able to adapt as the patient's abilities change. Data collection modalities, utilizing different modalities like mobile apps, electronic clinical outcome measures, and web-based platform. Digital biomarkers, and we briefly touched upon by Adam on how we can utilize objective biomarkers in collection of gait and balance in individuals which have cognitive decline. Remote monitoring and telehealth, utilized electronic clinical outcome measures utilizing technology for remote patient monitoring and telehealth visits. And regulatory advances, making sure that we take into account any changes in the regulatory guidance looking into data security measures like blockchain technology, and explore new technologies for data analysis. And that could be, using visualization tools, making sure that it is the endpoints are analyzed statistically and with a clinical with a clinical input. So with that, I'll hand it over to my colleague, Dr. Patel.
Thank you, Dr. Verma. So I'm going to talk about the considerations for cardiovascular disease and cardiac safety monitoring in patients with Alzheimer's disease in a clinical trial. We can go to the next slide. So the link between cardiovascular disease and dementia has been known for quite some time, and it's known as the heart-brain axis. It's been supported by large observational cohort studies and data from experimental studies. But it really isn't surprising that there's a connection between these two diseases, since the risk factors that are involved with cardiac disease and dementias are actually very similar. There's also clinical data from some trials that show serum biomarkers also support this connection. In one specific recent study, the levels of amyloid beta, which has been considered as one of the mechanisms for Alzheimer's disease, were higher in patients that had cardiac disease and dementia, Alzheimer's disease. And in patients that did not have cardiac disease or Alzheimer's, there were lower levels of amyloid beta level in the serum. We can go to the next slide. So as I mentioned, a lot of the risk factors are common between cardiovascular disease and hypertension. They include many of the risk factors that we think of with cardiovascular disease, such as hypertension, hyperlipidemia, diabetes, metabolic syndrome, and cigarette smoking. Pathologic studies have also shown that subclinical development of atherosclerosis of the carotid, femoral, and coronary arteries is associated with development of dementia and Alzheimer's disease. There's also strong genetic evidence that links the two diseases. Specifically, polymorphisms in the ApoE4 gene are associated with higher risk of developing Alzheimer's disease and cardiovascular disease. And the molecular dysfunction, things like chronic inflammation, oxidative stress, and atherosclerosis are also associated with the development of both cardiovascular disease and Alzheimer's disease. We can go to the next slide. Now, this is a figure from a recent paper in the Journal of the American Heart Association, and it shows the overlap between the risk factors for cardiovascular disease and Alzheimer's disease. On the left-hand side of this figure are the risk factors for Alzheimer's disease. On the right-hand side are those for cardiovascular disease. And you can see in the middle, in the blue circle, there's many risk factors that overlap. Many of those we just discussed: genetic variants, hypertension, hyperlipidemia, depression, lower socioeconomic status, oxidative stress, and chronic inflammation are just some of them that are overlap between the two diseases. We can go to the next slide. Now, one of the really important things to consider and keep in mind with Alzheimer's disease and cardiovascular disease is the temporal relationship between the two diseases. So while there is an overlap of the two diseases, clinical cardiovascular disease precedes the development of clinical dementia or Alzheimer's disease by at least a decade. And the mechanisms by which this occur aren't completely understood, but obviously one of the reasons that this might happen is that when you develop cardiovascular disease, you now have poor cerebral perfusion and the development of neurovascular disease, which can then exacerbate or even induce dementia. The fact that cardiovascular disease clinically precedes the development of onset of dementia or Alzheimer's disease means that almost every patient with Alzheimer's disease will have underlying cardiac disease. And this is the important and critical thing that we need to keep in mind. In fact, when you looked at age-matched controls, patients that have Alzheimer's disease have much more severe cardiovascular disease than similar patients of the same age when they don't have Alzheimer's disease. There's also an independent association between several specific cardiovascular diseases and molecular dysfunction, such as chronic inflammation and oxidative stress. These include atrial fibrillation and coronary artery disease with Alzheimer's disease, where we know that these molecular dysfunction are risk factors for the development of these cardiovascular diseases. We can go to the next slide. So with regards to atrial fibrillation association with senile dementia and Alzheimer's disease, A recent paper was published that showed data from the Intermountain Health Consortium. In this study, 37,000 patients were enrolled and prospectively followed for 5 years. The patients had a mean age of about 60 years old, but over the 5-year follow-up, 27% of the patients developed atrial fibrillation, and about 4% of them developed Alzheimer's disease. And what was seen here is that the patients that had dementia and Alzheimer's disease were older, had more heart failure, had more diabetes, had more renal failure, more stroke, and more coronary artery disease. But in addition, there was also a higher risk of developing AFib independently with Alzheimer's disease in this study. And that risk was higher for patients that were younger than 70 years old. Most importantly, surprisingly, along with this association of Alzheimer's disease and atrial fibrillation, patients with both atrial fibrillation and Alzheimer's disease actually had a 45% higher risk of mortality compared to Alzheimer's patients that did not have atrial fibrillation. We can go to the next slide. Now, this is a study that shows the association between coronary artery disease or coronary heart disease, as is known in some parts of the world, and dementia and Alzheimer's disease. So this was done in a retrospective analysis in the UK Biobank. Now, the UK Biobank is a very large biobank with about 500,000 subjects from the United Kingdom. They have clinical data, electronic health data, biomarker data from serum, imaging, genetic data. And so it's a very large database that's been mined for looking at a lot of association studies. When they looked at about 432,000 patients in UK Biobank for an association between coronary artery disease and dementia, they found that the patients had a mean age of about 57 for about the 12% of patients that had coronary artery disease in the UK Biobank. And what they found with this is that for every 10 years that the patients were younger when they developed coronary artery disease, there was a 25% higher risk of developing dementia. When you look at Alzheimer's disease, for patients that got coronary artery disease for every 10 years younger in age, the risk of developing Alzheimer's disease increased by almost 30% for every 10 years that they were in age. So this goes along with the fact that the preceding development of coronary artery disease or cardiovascular disease will often precipitate, but then even lead to the development or association of dementia and Alzheimer's disease. We can go to the next slide. So finally, I just want to leave with you some current recommendations of how to monitor cardiac safety in patients with Alzheimer's disease in your clinical trials. So total ECG should be collected in all phases of development, phase 1, 2, and 3. And you should be looking specifically for changes in ECG intervals from baseline during treatment and for new morphological changes, specifically those that might be indicative of new ischemia or myocardial infarctions. Blood pressure is something that should be monitored for all phases in patients and during drug development for Alzheimer's disease. About 70% of Alzheimer's patients have underlying hypertension, but you also want to look for exacerbations and changes that might be acid related. So in the early phase, the goal is to really do real large changes in blood pressure. And you really would be using office blood clinic assessments in this setting. Automated office clinic assessments can remove the operator and white coat hypertensive effect. And this is something that's been outlined in the draft FDA guidance for measuring blood pressure in clinical trials. In later phase trials, the goal is to rule out small changes in blood pressure, less than 3 millimeters of mercury, because as you're getting chronic therapy for Alzheimer's disease, you want to make sure that you're not inducing or increasing blood pressure, which could then lead to an increase in cardiovascular disease. 24-hour ambulatory blood pressure is the modality that's recommended for in late phase trials to rule out small blood pressure changes. This can help you assess both daytime and nighttime blood pressure changes, as well as diastolic and systolic blood pressures, as again outlined in the draft FDA blood pressure guidance. Home blood pressure can also be used in late phase trials to get more dense data for monitoring for unscheduled visits, or if there tends to be a mechanism of preclinical data where there might be a higher signal for blood pressure changes, this can be added as well. And then finally, as we mentioned, Alzheimer's patients do have atrial arrhythmias and atrial fibrillation. So continuous ECG monitor is something that should be considered in trials so that you can know that the patient, what the baseline data is for the incidence of AFib in these patients, since they have so much, begin with and make sure that you're not inducing the treatment related. This is important to consider because with continuous ECG monitoring, you collect such large amounts of data that it's important to get sufficient amount of pre-dose data and have appropriate placebo-controlled patients in these studies so that you can discern on-treatment changes or development of atrial arrhythmias from those due to the pre-existing clinical disease. And then finally, I didn't mention this here, but cardiac imaging also has a role in Alzheimer's disease. This is mostly to assess cardiac structure and function prior to enrolling patients in trials, because many of them will have LV dysfunction or abnormal cardiac function. With that, I'm finished, and I'll turn over back to Dr. Rudo to give us some concluding remarks.
Fantastic. Well, a sincere thank you to all the panelists for your contributions today. As promised, quite a packed session here, dense with considerations for digital endpoint strategy in an Alzheimer's development program. You've heard about the role for PET and MRI in confirming eligibility as a biomarker to determine efficacy, and the importance for imaging for the development of ARIA, that safety risk for in amyloid therapies that Chris mentioned. At first pass, it may not have been obvious why we were talking about movement and gait analysis and what is classically thought of as a memory disorder. You now have a better understanding, I think, for how cognitive function does impact gait and balance, how this could be leveraged as a biomarker in the drug development program. From an ECOA strategy standpoint, considerations for optimizing the platform you deploy for the unique challenges of Alzheimer's patients, the critical role for rater training, how this can impact the precision of the data collected. And regarding cardiovascular risk, Dr. Patel just took us through how to manage the high risk that these patients often bring into a clinical trial. This is important not just in understanding that those cardiac risk factors are associated with Alzheimer's, but also proactively thinking about how to manage those risks during study conduct. In particular, developers need to consider the risk for false positive safety signals, for example, that may be attributed to drug if a sufficient baseline test strategy isn't deployed. So with that summary, we're going to transition to a brief Q&A in the last 5 minutes or so here. Please continue to use that chat box to enter your questions. We've had a good number come in already, and I'll certainly start there. I'll invite the panelists to all come back on screen. And for any of the questions we don't get to today that you've submitted, we'll commit to directed follow-up with you offline, so you can have those answered. So the first question here is directed to Adam regarding the presentation on gait analysis, and this looks like a topic you certainly covered to some degree, and I think maybe just some clarity. Could you explain further why we'd want to consider wearable sensors to measure gait and balance in an Alzheimer's program versus just focusing on more common imaging endpoints only?
Yeah, sure. Thanks, Todd. So I guess in addition to the evidence I was showing and some of the models I showed in the slides, there's some evidence linking changes in gait, specifically pace and rhythm with measures of neurodegeneration. And more so, the studies additional studies are showing that this analysis, this type of instrument analysis is feasible, and patients accept the technology, and these outcomes are meaningful to patients. So there's an important aspect that can be related back to patients' perspectives. And these are relatively easy to implement. This technology is relatively easy to implement in trials, larger trials. There's a short setup time, relatively short test duration. So these are all benefits in that sense. So all in all, I think there's good promise using this type of instrument and analysis as a clinical tool for screening for disease progression and supplement to traditional imaging and clinical outcomes.
Great. Yeah, that's helpful. Thanks. Chris, I'm going to direct this one to you. You went through quite a few different imaging modalities here. Can you explain further how we should know what is most sensitive for a specific development program to avoid excessive testing, which of course is both expensive and inconvenient for patients? So picking modalities is the theme here.
Yeah, sure. No, thanks. And that's a fantastic question. That my answer can vary here pretty substantially depending on the compound, the design, the cohort, and even the phase of, any potential trial that's coming in. As we've shown and discussed greatly, safety is obviously a must-have for ARIA detection and other serious adverse, events. So I certainly think that we would recommend more advanced functional exploratory endpoints and try and substantiate and try to, really glean any sort of therapeutic efficacy throughout the lifespan of the trial. Certainly statistical testing and correlations with clinical outcomes will ultimately help drive the decision for what to include. And I think, not to plug or anything, but I think this is exactly where, we at Clario, particularly in the neuroimaging group, can help decide and help demonstrate efficacy within your trial.
Great, thanks.
Sure.
Dr. Verma, this one is directed at you regarding sort of newer eCOA endpoints. So are there any evolving or newer eCOA endpoints you've seen being developed now or even starting to be used in recent Alzheimer's studies?
Yeah, that's a great question. And to answer that, we ran a clinicaltrials.gov. We ran a bot to analyze how the AD landscape is changing. And what we did come across was that given that we are seeing more and more studies being designed in MCI in early stages of AD, particularly focusing on stage 3, there is also guidance from FDA as well to look into integration, of instruments or domains. Conceptually, as in the case of CDR, Clinical Dementia Rating Scale, we do see that we can have a unitary construct. So what I mean by that is we can have both cognition and functional. But statistically, what we are seeing is that by combination of separate measures like ADAS-Cog with ADCS, ADL, we are seeing a shift towards using composite measures like ADCOMS and IADRS. So, these are some of the instruments or perhaps composite measures being employed in the new programs that are being designed.
That's great, really helpful, thanks. Dr. Patel, we're going to squeeze one more in here for you. This question came in actually as you were on your last slide, so you touched on it, but maybe can elaborate. You mentioned ECG and heart rhythm monitoring. Is there a role for cardiac imaging like echo to assess safety in patients with Alzheimer's disease? And if so, when should this be included?
Yeah, thanks, Todd. No, that's actually a very good question, Dr. Rudo. So as I mentioned, we really recommend that you're going to get at a baseline and screening, right, to make sure that the patients don't have significant underlying heart failure or dysfunction. Since most Alzheimer's disease therapies are not going to be cardiotoxic or based on monoclonal antibodies or neuropenetrant, you don't really have to have a lot of cardiac monitoring, in these patients if you've ruled out significant disease at screening. However, if there's a preclinical signal or if the modality of the asset tends to be maybe looking at something that might be cardiotoxic, that may be where we want to include on treatment monitoring, maybe every 3 months or so, depending on what kind of signal you're seeing.
Great, thank you. All right, well, it looks like that's all we have time for in terms of Q&A, but as mentioned at the start, we'll happily follow up with any of you individually who submitted a question, but we didn't get a chance to get to today. Ryan, I will turn back over to you to close us out. Thanks.
Well, thank you all of you for this very insightful discussion. We have reached the end of the Q&A portion for this webinar. If we couldn't attend to your questions, know that the team at Clario will follow up with you, or if you have some further questions, you can direct them to the email address up on your screen. For those interested in hearing more from Clario, you can register for their next webinar, Revolutionizing Ophthalmology Clinical Trials Through Enhanced Accessibility and Advanced AI-Driven Analysis. And this is scheduled for Thursday, July 11th, and you can use the link that I'm sending now in the chat box. Now, I want to thank everyone for participating in today's webinar. You will be receiving a follow-up email from XTalks with access to the recorded archive for this event. A survey window will be popping up on your screen as you exit, and your participation is appreciated as it helps us to improve our webinars. Additionally, I've also shared a link to view the recording of today's event in the chat box, which you can also share with your colleagues so they may register for the recording here as well. Now, please join me once more in thanking all of our speakers for their wonderful time here today. We hope that you found the webinar informative. Have a great day, everyone, and thank you for coming.
Thanks.
Thank you.
Thank you.
Thanks.